We are very happy to have Professor David Nelson from Harvard University as our speaker for today. David got his PhD in physics from Cornell University in 1975 under the supervision of Michael Fisher. And then he became a junior fellow in the Harvard Society of Fellows. Since 1978, he has been a professor at Harvard and is now the Arthur K. Solomon Professor of Biophysics, Professor of Physics, and Professor of Applied Physics. Much of David's work focuses on statistical physics, geometry, and topology as applied to soft condensed matter physics. And among his many achievements is the theory of melting into dimensions developed together with Bert Halperin and predicted the new partially ordered intermediate phase between the solid and fluid phases. This phase, that's known as the hexatic phase, was directly observed in several experimental systems, including a direct observation in 2D colloidal monolayers as reported 3 years ago. And in recent years, David extended his research work towards biophysical systems, among which is the topic of today's talk, titled On Growth and Form of Microorganisms on Liquid Substrates. So David, thank you for accepting our invitation. The virtual stage is yours.
Well, thank you very much. It's an honor and a privilege to speak at this series and to at least be able to interact with my many friends in Israel and in other places in the world. And what the motivation for what I'll tell you about is that, as many of you know, life probably first evolved in a liquid environment. And in particular, water played a major role, covering a large fraction of the Earth's surface for most of our history. And there's evidence for, fossil evidence actually, for oxygen-producing cyanobacteria. There's an example over here in the upper right, Senecacoccus, that goes back maybe 2 billion years. And these bacteria, notice they're green, transformed our world because they invented photosynthesis. In fact, these Senecacoccus have a common ancestor with the chloroplasts that eventually were incorporated into green plants. And their spatial growth and evolutionary competition, also many other organisms in the ocean or in lakes and ponds, occur, of course, in a liquid environment. And this can happen at both high and low Reynolds numbers. These photosynthetic organisms control their height to resist downwelling currents. They have to stay close to the ocean or lake surface because they need the photons to do their photosynthesis. So this is the motivation. Here's a picture of a bloom of cyanobacteria over a lake in Guatemala. Notice these are striated. They're not mixed to uniformity, at least not at this moment. And I'll try to argue that there's good reasons why they never get mixed to uniformity. It doesn't actually behave like cream in your coffee, for example. Speaking to an audience which has many people from Israel, I know you don't need any encouragement to interrupt with questions, but please do interrupt, and I welcome people stopping me. There's plenty of time, and I don't have to cover everything, so please do interrupt. Here's another picture, actually off the southern coast of Iceland, of a plankton bloom. It's quite complicated, swirly pattern here in northern Atlantic. Here's a scale of 110 kilometers. And with those very large length scales, many of you will not be surprised to learn that there's a Reynolds number that controls what's going on here at the ocean surface. It's stirred up by wind-wave interactions, perhaps. And the Reynolds number is a dimensionless number that characterizes the Navier-Stokes equations that are advecting these organisms around, can easily be 10 to the 8th or 10 to the 9th. And that means there's a cascade of energy scales and length scales and time scales. And in typical oceanic turbulence, there's a large eddy turnover time, maybe 2 months or 50 days. There's a cascade of eddies that goes down to the smallest scale. Most of this is happening in the first 50 meters of the ocean.
David?
Yep.
David, can I take you up on your offer?
Sure, please. Please, Dov.
Should I think of this as being mainly two-dimensional, or should I think about it being three-dimensional because it modifies the cascade?
Yeah, so thank you. It's quasi-two-dimensional, and I'll show you a cartoon which illustrates that. The photosynthesis happens in the first 50 meters or so, because photons can't go that deep, and the air currents and the wind that stir up the ocean will typically produce agitations that go roughly that depth. So it depends on how you coarse grain, but as you'll see in a minute, if you coarse grain on a scale of 50 meters and larger, most of the action is quasi-two-dimensional, and that'll be important, as you'll see. So, other questions? So thanks, Dov. The other thing is that the important time scale, in addition to all these eddy turnover times, it's 50 days down to 5 minutes, and every time in between, basically. It's a whole cascade of time scales. The other time is, say, the plankton or Seneca caucus doubling time, and they typically reproduce every 12 or 24 hours. And so their doubling time is typically in the middle of this cascade of eddy turnover times, and they can, for the large, slower eddies, they can adjust their behavior to compensate or adapt to what the eddies are doing. Well, that's sort of a problem that I've been interested in for a while. There's a simple way of beginning to get a handle on this is to take some turbulent velocity field, say some model of what's going on in a slice of the ocean near the surface with a velocity field u, and then let's see is the concentration of these organisms in space and time. There's advection, of course. They may diffuse around if they have flagella and are sort of swimming randomly. There'd be an effective diffusion constant. But there's also this logistic term, which is involves the growth of the organisms at a rate mu associated with their doubling time. And then it's there's some saturated carrying capacity. Let's assume for the moment we have abundant nutrients. The important aspect, another important aspect of this, which is different from just diffusion of a passive scalar, which you might, an equation you might write down to describe pollutants coming out of a smokestack, for example, in addition to this logistic term, is that typically these, the effective 2D velocity field, if I coarse grain, as I was talking to Dov about, the effective 2D divergence is going to be non-zero. This is a cartoon that summarizes many of the complicated processes going on in the ocean. And there can be downwellings. It's a three-dimensional incompressible fluid, typically. But if the organisms control their buoyancy, and most of them do if they're photosynthetic, they will resist these downwellings. And so if the three-dimensional velocity field looks like what I've shown here, what will happen in effect is the organisms will get compressed locally and rarefied where you have upwellings, compressed for the downwellings, rarefied for the upwellings. So effectively, you have a non-zero 2D divergence. So we have an interesting problem where we have both organisms that are not pollutants, they're dividing, they're growing, and in various ways, and an effectively incompressible velocity field. And that's a somewhat of a new game in high Reynolds number turbulence, for example. Here's a simulation that was done by Prasad Perlikar and Federico Toschi and my collaborator, other collaborator, Roberto Benzi. And what all this does is solve the equation that I showed you earlier with a velocity field that is At the time, I think it was sort of state of the art for high Reynolds number turbulence in 3 dimensions. So it was incompressible turbulence in 3 dimensions, but we only allow the organisms to live at the midplane of a periodic cube, which had turbulence with a Reynolds number of about 100,000. And so the effective projected velocity field, so the idea is they control their height to stay at the midplane of the cube, so they can only move in that plane. This is what happens in the plane. And it really is quite different than our intuition from stirring cream into our coffee, because it never settles down. It's a statistically steady state, but these organisms are constantly growing up. As they grow, though, they're being compressed. They can be compressed to larger than the carrying capacity, which rescaled to be 1 here. So this becomes negative. Then they die out because there's not enough food. And there's this interesting statistical steady state associated with this equation and a turbulent compressible velocity. And you could have lots of fun thinking about things like—
Excuse me, David, can I ask
Yes,
a question? The 2-dimensional flow in the second equation, is it the solution of the first equation, or the equations are solved simultaneously?
Thank you. There's no feedback in this model of the growth and saturation, et cetera, of these organisms back onto the Navier-Stokes equation. We assume that they're sufficiently dilute. So what happens is they are in a sense solved simultaneously, but this first equation affects the second equation. And the way I generate this U2D is to solve the full 3D Navier-Stokes equations, or rather my collaborators did, and then take that solution and just look at the projection of the velocity field onto a plane. So we ignore the vertical component of the velocity field at the midplane of the cube. So they are essentially solved simultaneously, but there's a one-sided bias where this equation influences that one, but not the other way around.
And the time scales of the diffusion coefficients, et cetera, are chosen to be comparable to experiments, or?
Comparable to organisms in the ocean. So the viscosity, which that's one time scale, is 10 to the minus 2 in CGS units. Diffusion constant is chosen to be typically Stokes-Einstein result for an organism that might be 10 to 20 microns across. If it has flagella, the diffusion constant gets amplified by maybe a factor of 1,000. This Schmidt number is typically small. There's, of course, a Reynolds number that comes into play. We'd like it to be large. We can't get it to be 10 to the 8th with current computers, but we can get it to be 10 to the 5th. And then the other dimensionless time, Yuriv, is the ratio of the doubling time. So this mu up to factors of log 2 is the reciprocal of a doubling time when the organisms are dilute, as they are maybe over here. And then you compare that to the small eddy turnover time of the turbulence that you're imposing. You get a sense of how fast the organisms can reproduce. And the interesting regime for most of this lecture, including actually the laboratory experiments, which will be at low Reynolds number, but nevertheless in the same regime for this time ratio, will be where the doubling times are small compared to at least some of the eddies in the system. That lets the organisms be able to reproduce and compete in response to what the fluid mechanics is doing. So I'm trying to actually engineer a shotgun.
David, can you say what goes into the force term in Navier-Stokes equation?
Okay, so yeah, so the force, thank you, Yakov. The force term in the upper right is basically, think of it as like a spoon in a cup of coffee, perhaps. It stirs the cube. It has Fourier components, spatial Fourier components at large wavelengths and time components that correspond to a large eddy turnover time. So it injects energy at large scales, and then these nonlinear pieces, a la Kolmogorov, ideas about turbulent cascades will couple things down to smaller and smaller scales until finally the viscosity will dissipate. So it's a large wavelength stirring and stochastic in time.
David, I also have a question.
Yes, please.
Yes, so the physical origin of the compressibility term, does it have to do with the fact that you have this inhomogeneity of the density of these organisms?
The organisms, thank you for the question. They certainly are quite inhomogeneous, as you can see in this movie. But the physical origin is because we're coarse graining a three-dimensional, we imagine coarse graining a three-dimensional incompressible velocity field in the ocean, let's say. But the organisms control their buoyancy, so they don't go down. And so effectively, it's like, I don't know if you know Walter Goldberg's experiments on floaters.
Right.
Polystyrene spheres at an air-water interface. When you stir, they also respond as if they were under the influence of a compressible 2D velocity field. And we're coarse graining on scales large compared to, say, 50-meter depth of where the turbulent activity.
But doesn't this fact couple the two equations actually, indirectly?
Well, it could. You could in a more sophisticated model. What we're doing, there's no organism dependence here. You could imagine that if the organisms were quite dense, they would feed back and produce forces in this Navier-Stokes equation, F equals ma, basically. But here we don't take that into account. And it turns out for the experiments that I will show you, that's a very good approximation. But of course you could look more carefully and see what's going on.
So may I ask also a question, David?
Please.
Why the Fisher velocity of the bacteria do not appear in this one of these dimensionless parameters? Because I would guess
go ahead.
The ratio between the Fisher velocity and the velocity in which these attractive regimes are moving that will determine whether or not you have localization of this type or not.
Yeah, that's a very good point. So what Nadav is suggesting is that in the absence of flow, there's a Fisher wave that's associated with this nonlinear equation here. And there are other dimensionless numbers, and with Roberto and Abby Plummer, who was a student of mine, we've looked at the dependence on that ratio. So this here are just three examples of what you could put here. Absolutely. Other questions? This is great. Okay, so with that motivation from the ocean, I also want to say you can all play other games, okay? So in particular, what we saw in the previous movie here is one species, but I could have a Darwinian competition between species, and I won't have time to go into it here, but with Andrea Giametto, who's now on the faculty at Cornell University, and my longtime collaborator in biology, Andrew Murray, and then Max Lavrentjevich, who is now at the University of Tennessee, we've thought about what happens if you have organisms that are maybe vected by fluids, but are antagonistic. And the antagonistic problem we find quite interesting because there are genetic wave dynamics generated by these interactions. And in particular, if you can make organisms as Andrea and Andrew have in the lab, which are so-called killer yeast strains, Saccharomyces cerevisiae, baker's yeast, basically, which produce toxins. And each of these yeasts will, in fact, make not only a toxin, but an antidote to the toxin. There's a toxin-antitoxin cassette in their genome. So they are immune to the poison that will kill their competing species. This is a budding yeast, so that's what the shape is. It involves a daughter coming out from some mother cell here. It's undergoing, let's say, asexual cell division here. And these toxin, NC-toxin cassettes lets you look at antagonism. And it turns out that although one of these may reproduce faster and produce more of the K1 toxin than the other, because of the antagonism, you have to rethink a little bit Darwinian ideas about survival of the fittest. If this magenta strain has a selective advantage over the blue strain, you'd think that it would take over, and it would in a well-mixed test tube. But if you have on a Petri dish, for example, no fluid flow for the moment, this magenta strain, only if it's bigger than a certain size will Darwin kick in and it will take over. If it's smaller than a certain critical size, the antagonism, the death and destruction because of the fighting at the interface wins out over the selective advantage, and below a critical radius, much like nucleation theory in physical chemistry, this will collapse, this little droplet will collapse. So that's another game you can play. Here's a set of reaction diffusion equations where we're love to put in a turbulent velocity field, but here we're just going to put in something very simple. It's a cellular flow. It's a steady flow given by these components. We can tune it to be compressible, incompressible, whatever. If it's incompressible, also I'll talk about incompressible just to illustrate with this example, this particular cellular flow here, which advects now two competing species, has, if you look at the corners and stuff, two saddle points, one positive circulation vortex, one negative circulation vortex, embodied in its periodic boundary conditions. And now I have sort of two coupled logistic equations, as well as diffusion, as well as advection. And the important parameters here that parameterize the antagonism are these epsilons. So these epsilons will tell you whether the growth rate goes up or down in the presence of the competing species. So you get a little boost, a boost if epsilon A is positive, but in fact, it's a diminution in the growth rate if you have this poisoning. And similarly here. And there's an antagonism parameter in the fluid mechanics, which is the average of these antagonisms. They're negative, so this is a positive number. And then this turns out that epsilon A minus epsilon B plays the role of a selective advantage, and we can actually look at this droplet physics or droplet science. And for this particular flow field, that's a whole nother talk on what actually happens in detail, but just to give you a sense of why we think this might be interesting, here's a droplet of, would have been the magenta strain, but now it's red. And it has a selective advantage, take my word for it, over this blue strain. I'm just solving those reaction diffusion problems. We can look at the fate of this droplet in this saddle flow. And you can see interesting things happen. The saddle flow tends to stretch it out. There's effectively a line tension, an organismic line tension between these two species. The whole plane is filled, it's incompressible. The whole plane is filled with these organisms. And then the droplet actually pinches off and breaks. And you could start off bigger than the critical radius, and this could be smaller than the critical radius. We have periodic boundary conditions, so things come in. And eventually, species A, the one with the selective advantage, does take over, it turns out, in this case, but it takes over by an extremely circuitous route. And the fluid mechanics can completely change what you might have thought of with a simple survival of the fittest perspective. Okay, so one last example, then I'll turn to the experiments.
And David, is the surface tension a heuristic analogy, or can you define something more precise?
Okay, very good, very good. Thank you, Yuriv. It's both heuristic and in some cases literally like a surface tension that we know and love. There's aspects of this problem which look like, oil and water droplets and so forth. But there are other aspects of it, in particular at the interface, which have to do with organisms dying and reproducing. And that's not something you would normally encounter in physical chemistry. And in particular, the nature of the noise, the stochastic effects are very different. The noise in this problem is number fluctuations, local number fluctuations, which I've left out of this particular simulation. And that's one area where the notion of the conventional ideas about surface tension, escape over a barrier, etc., have to be modified. And we still don't completely understand how that plays out. This was discovered by a very insightful population geneticist named Nick Barton. And he was a field ecologist in graduate school, and he read Sidney Coleman's lecture notes on the fate of the false vacuum and actually figured out how to apply it to this class of problems. Not with the fluid mechanics, but just for ecological problems. Other questions? All right, one last example, I'll just mention it very briefly. We don't have to, this is where the number fluctuations that I mentioned to you, Reeve, come in. If I have a compressible turbulent flow, and this is again the same kind of flow that I showed several slides back, I can have organisms which are advected and reproducing in this flow. And so we have green organisms and green squares and red squares. And if you look carefully at these things, they are actually dividing and competing. They're competing in a neutral sense. They are reproducing at the same rate. They're not making toxins, although we'd like to put that in and see what happens. And the compressible turbulence greatly reduces the carrying capacity, reduces their numbers. The population never goes extinct in the simulation, but eventually, if you run it long enough, one or the other of these will take over. In fact, if I go out to large times, I think you'll see that the red has triumphed. But if you do it with random initial conditions, they're neutral, and you do it with 50-50 initial concentrations, half the time red will win, half the time green will win. So these are the world lines of these organisms, and this is cell division, and then this is death. Could be either color coming in here. When you get too concentrated in our agent-based simulation, one of the two organisms will die. So that's just motivation. And would be nice not just to do simulations, not to make just inferences from satellite photography provided by NASA or some other agency of plankton. It would be nice to have simple reproducible laboratory experiments and to explore, in my opinion, these fluid flows and see how they affect spatial population genetics. We're not going to easily get high Reynolds number in the laboratory, but low Reynolds numbers are also quite relevant biologically. Depends on the situation. And the question is, can we impose flows with upon organisms with reproduction times that are much less than a typical eddy turnover time? So the organisms can divide many times as they swirl around on a particular eddy. And so for a while we thought we'd play around with pumps and syringes and see what we could do in the lab. So I'm going to tell you in the rest of this talk about the results of that exploration. It was published about a year ago, and this is where the growth and form of microorganisms on liquid substrates come in. These are some of the questions. Yes, Erez.
Yeah, hi. Did anybody try elastic turbulence as a medium to
I don't know. that anyone has tried, it would be quite interesting to do that. Absolutely. It would be very interesting. The crucial thing that I'm going to focus on, Erez, is I want to look on timescales that are large compared to the cell division times. So whatever, the class of problems, maybe elastic turbulence could be an example, is where we have growing, reproducing organisms taking over, spreading out over spatial territory. But I don't know of anyone who's done elastic turbulence. Thank you for the suggestion.
Okay, thank you.
Anyone else? So here are some of these patterns. This is all in a Petri dish where the organisms are floating at the interface of a liquid. It's a little bit like Walter Goldberg's floater particles, but these are alive and they carry a heritable genetic marker, say red or yellow or green or red. And you'll see that these yeasts stir up the fluid on which they are floating. And they do it without flagella. These are yeast experiments that I'm going to tell you about. The people who did the hard work are here. This is Severine Addis, who's now a postdoc with William Irvine, I think, in Chicago. This is Brian Weinstein, a very talented graduate student. This is my colleague in molecular and cellular biology, Andrew Murray. So this is what I want to tell you about the remainder of this presentation. So turns out we didn't need pumps and syringes. These organisms, even though they don't have flagella, they don't have any intrinsic motility, spread across the top of the liquid by creating their own flow. And so here's an old experiment, so simple that even I could do some of these experiments with yeast, starting off in this homeland, spreading out radially. And this is on a hard agar plate, so there's no flow right now. Flow is about to appear. In this little halo region, this is kind of an inflationary dynamics as you expand radially in 2 dimensions. In this little halo, the red and the yellow are fighting it out. They're trying to decide as they grow, as they reproduce at the frontier. Here's the current frontier of this range expansion. Who's going to win? And what happens is a stochastic outcome locally at any angle or position around this growing colony. It's either going to be yellow or red. And it decides due to number fluctuations. So the stochastic effects that Yariv was alluding to play a role here. If I now just take this experiment and redo it with this homeland, the same size inoculant approximately, in the center of an agar plate, there's an epoch of genetic demixing. That's just this thing here, but it gets stretched out on a liquid. Now it's a very viscous liquid. This liquid has a viscosity of about 600 Pascal seconds. That's about 600,000 times bigger than the viscosity of water, but it's still a finite viscosity. And something new is happening in the early stages of this range expansion before it decides to be red or yellow. And the take-home message, which I'll go through in detail, is shown down here schematically. When this sort of thing was happening on liquid media at the interface, these are the yeast sit at the interface of the nutrient broth and air over the entire time scale of the entire time course of the experiment. What happens is they generate, for reasons that I'll try to explain, a submerged vortex ring underneath the colony. And we detected this vortex ring by putting in fluorescent beads. These are not reproducing beads here. These green things are, let us do particle imaging velocimetry, and I'll show you a map of what's going on in the fluid mechanics underneath this colony. And if the colony's not there, no vortex ring. So the colony itself, without pumps and syringes, generates flows that dilate the growing cell mass, then extra dilation above and beyond the simple reproductive growth. There's a kind of a Fisher wave here over the hard egg, on the hard agar substrate. Here it's something very different, which I want to tell you.
So, sorry, question. I missed something. If you take yeast and put it in a culture dish or something, they sink down. You get everything on the bottom.
Yeah. So thank you.
Why here it's
okay. Yeah. So we've looked. The yeast are, even when I mix in, make this very viscous and mix in cellulose or whatever we do to control the viscosity over here, Aris, the yeast will eventually sink down, but the real physical surface tension keeps the yeast floating at the interface. We've looked carefully with microscopes and so forth on the time scale of the experiment. So the colony never sinks over, say 7 days. It just stays at the interface, and but if you waited long enough, it would sink down. And I'll show you experiments where we actually nucleate a colony on the bottom. So
this is the budding yeast, or
yes, it's budding yeast, Saccharomyces cerevisiae. And in fact, if you look in the early stages, eras, you can see after you inoculate this homeland here in the middle, that the yeast are attracted to each other by capillary forces, like Cheerios floating on milk in a cereal, and they coarsen even before they divide due to that mechanism. And the capillary forces keep them there, just like, as you know, capillary forces can keep insects, for example, from sinking into water.
Thank you.
Yeah, sure. Other questions? All right, so here's a more detailed example of what's happening here. This is this colony that forming with this mottled mixture of blue and yellow genotypes. I'll let it grow here. I'll let it grow outwards, so let it grow like that. And out here at the edge, you can see it finally decides by reproducing with this heritable genetic marker that it's going to be blue or yellow, okay? And, but notice, look inside this red circle, there's an object there that to us, or at least to me, looks like a lizard. Here's its tail, here's its head. And let's follow the lizard back in time as I go backwards and see where the lizard came from. And if you look, this lizard has the same shape. The colony is undergoing a conformal angle-preserving transformation as it comes out. So let's just let this go again, and all these shapes, until you get to this final stage here where you decide to be blue or yellow, all these shapes are preserved. So there's some angle-preserving map that's produced by the reproductive dynamics of the yeast at the viscous liquid air interface. You can get some understanding of what's going on by just doing a simple fluid mechanics calculation where we have a very viscous yeast liquid having all the nutrients the yeast need, has a thickness of maybe 1 centimeter. This is a standard, roughly a standard diameter of a Petri dish. And then there's the velocity field. There's a liquid down here. And then treat the colony itself as a 2D liquid up here floating. The colony thickness is maybe 200 microns when it matures. And then the simple model is basically looking at the density, the 2D density of the the two genetically labeled organisms. They are neutral in this particular class of experiments, but they could have some antagonism or something else going on. But in any event, their density obeys the equation of continuity, of course, in terms of this 2D velocity field here. But there's an additional source term because there's cell division. And so if rho 2D is the cell density projected down for this colony, then you can solve this equation, and the divergence of this 2D velocity field is actually the growth rate. So here's another source of effective 2D incompressibility, just the growth itself. And you can find that there's indeed a radial velocity field of this colony just because the organisms are dividing with excluded volume interactions and pushing each other outwards once they reach this height. So, okay, questions? This radial...
So I have a question.
Yes, please, Aris.
So it's a very thick layer compared to the size of the cell itself.
Yes, correct.
Many cells inside this layer,
yep.
If it's not mixed, will not get food, and you'll be stuck from division by their neighbors. So how much of the colony is really dividing it?
Yeah, thank you. So these yeasts are about 5 microns across. So as you say, the colony is about 40 of these yeast cells thick. And people have, Pascal Hertzen and Andrew Murray have studied how efficiently the wetness of the medium is siphoned up with its nutrient to allow this colony to grow. And as far as we can tell experimentally, it's consistent with those experiments. They're all dividing roughly at the same rate. There may be lesser divisions up here when you get so thick that you can't get the nutrients. But that nutrient limitation is basically what determines this thickness H. So it's about, as I said, 40 cell widths thick.
Thank you.
Right. In any case, this radial velocity field preserves angles, and you can understand some aspects of the stretching. And now the radius of the colony itself and the radius of this lizard-like figure are increasing exponentially in time. In the early stages, you don't get Fisher waves. All the nutrients, turns out that submerged vortex ring helps bring in the nutrients from the far corners of the Petri dish. So they're all dividing. And again, this addresses a little bit of Aris's question. The radius and all the dimensions at this period of time are increasing exponentially. So we sort of have the Hubble constant in our little inflationary universe of spatial population genetics. Okay, and so here's more detail. There's something more going on, though, than this simple pushing. This is the submerged vortex ring, and this is our measurement of the velocity field. This is the colony right here in the darker blue, and this velocity field is extending way out beyond the colony radius, maybe by a factor of 2 or more. Here's an azimuthally averaged velocity field. Velocity field falls off eventually when you get far from the colony at this stage. You can understand it, this extra stretching, which is what happens when you have the submerged vortex ring. You can understand it by thinking of, here's a cross-section of showing the vorticity. And then there's because you have boundary conditions where you want the velocity field at the interface to be parallel to the air wall space, then behaves like anti-Helmholtz coils, it turns out. And in fact, there's an analogy with the magnetostatics that's illustrated down here. No magnetic monopoles is like incompressible 3D velocity field below the surface. etc. And you can show that this, this configuration of the submerged vortex ring gives another radial contribution near the center of the colony that stretches the colony outwards. Okay, so there's another stretching component due to the submerged vortex ring. I haven't told you where it comes from yet, but if it's there, it's going to produce some stretching. Okay, so what's going on? And when we first got these flows, some of my distinguished fluid mechanics colleagues said, "Oh, you guys are just somewhat careless. There's temperature gradients, and maybe there's a Rayleigh-Benard instability you don't know about that has nothing to do with this colony dynamics," which is a very reasonable question. But we redid the experiment. The Severine, Addis, and Brian are pretty good experimentalists, and they're pretty sure they eliminated the temperature gradient. And then if you redo everything with the same conditions but no colony, there's no flow. So the colony somehow seems to be causing the flow. Then there's the possibility there's some Marangoni physics going on, that the yeast is secreting, it may well do this a little bit, secreting some kind of surface active molecules that will push, change the surface tension, for example, and push the colony out. So we're worried about the Marangoni effect, which typically happens at, say, an air-water interface. So my collaborators, they redid the experiment. They grew the colony on the walls. They grew the colony on the bottom. And so now there's presumably no Marangoni effect. In fact, there was a submerged vortex ring in this case, but this one tends to compactify the colony. But then something else happens also when it's at the bottom that's actually a valuable clue to what happens when the colony is on the top. I want to show you with this movie. So this is a movie where you're looking down on a submerged colony. These green dots are not yeast, they're just the marker particles, so we can see what's happening with the flow. And if you look in the early stages, you can see the green dots moving, so you're getting some kind of flow. But then this happens. Any suggestions from the audience about what this is? Those are bubbles of carbon dioxide. In fact, we all know yeast is used to make bread rise. It's used to make beer. It does many good things. And down here at the bottom, there's not much oxygen, so there's some anaerobic metabolic pathway going on. This is one of these metabolic pathways that we force our biology students to memorize sometimes.
For yeast, it doesn't matter. For yeast, even with oxygen, they will ferment.
It will do something, indeed. You know better than I, Aris. It will do similar things with and without oxygen, but they're not identical. But the only thing I want to use this little experiment, we didn't pursue it further, to illustrate is that by metabolizing, that metabolism is important. Let's just put it that way. Here's kicking off the CO2 and so forth. If we now put the colony...
David, can I ask a question?
Of course, please.
Hi, I'm Gillian from Tel Aviv. So if you go back, so stressing on the thermal gradient point that you made, metabolism, I mean, produces heat. So how do you eliminate the heat being part of the...
Very good question. Metabolism does, thank you, Julie. It does produce heat. And let's talk about the experiments at the top. And we put in numbers to try to estimate what kind of temperature gradients might be present up here. It turns out they're pretty small, and we don't think they account for the experiments. Also, if it's hotter here, then it would be a little less dense underneath the colony, and that's a stable fluid mechanical situation. There's not, if it were heated at the bottom, then you might get a Rayleigh-Benard instability.
But you have to worry about lateral temperature gradient because there's no threshold.
Yep, absolutely. And it's possible that the thermal energy might be playing a role, but there's another effect, which I'm just about to get to, which might, which has this, goes in the same direction. And we think at least partially accounts for what we're seeing. It's possible thermal gradients play a role as well, but I'm going to argue, just to jump ahead, that density gradients are going to be happening here as well. Okay, not only am I going to get heat here, here's the metabolism that might be dominant, the more aerobic metabolism at the top. Again, Erez and probably others know more than I do about the details. Notice it's making ethanol, which is a good thing if you like beer. Of course, what we want to do, and Julie and Erez have anticipated this, is solve the Navier-Stokes equations in the presence of whatever is happening here. And heat could play a role, but there's also density. Why do I say that? After you run the experiment, if you let the colony suck all the nutrients out of this YPD liquid, you'll find that it's about 1.5% lighter than it was before you let the metabolism take place. And so, and you could probably provide a very similar analysis of what happens with the heat. So it's probably similar. We will be working in a Boussinesque approximation here, where we imagine that the medium density depends on the concentration. In this case, the concentration of nutrients. So nutrient could be driving it, possibly temperature. And we solve these two coupled equations with a gravitational field. And then, as Erez said, lateral effects are very important. And so what I'm showing here is a map, a kind of a weather map of what's happening below this growing colony. And if I say that for the moment there's no flow, but I'll ask whether flow could be generated by some sort of instability in a minute. Here I have the colony edge. These green lines are isobars, the usual constant pressure isobars you'd encounter if you dive to the bottom of the deep end of a swimming pool. But the red lines are the lines of, in this case, density of the medium, because there's a gradient because the colony is sucking the nutrients out and producing a lighter fluid. So the fluid gets denser and denser as you go down. These lines are curved. Okay? And here's the boundary conditions for how we suck in and out the nutrients. If you take the curl of the Navier-Stokes equations, you'll get this interesting result here. So turn on the laser pointer, the fake laser pointer. This is the vorticity. And I want you to focus on this term here, which arises when you have gradients in density, possibly also gradients in temperature. And we're at very low Reynolds numbers, so I'm not going to worry about vorticity advection or vortex stretching. Here's the viscosity. And what I have is essentially a vortex mill, a vortex generator, where when this cross product is non-zero of these gradients, which it surely is in this non-equilibrium situation, you can work out with the right-hand rule, this cross product, and you'll see that the vorticity that's generated swirls around as indicated by these arrows. It then diffuses away perhaps a little bit, but that we think is the source, one of the sources, probably the primary source of the vortex ring. If you put in numbers, the temperature gradients seem to be much smaller than the density difference that we measure. And so this is well known. Again, people in the audience probably know better than I. In atmospheric sciences, it's the baroclinic instability. Whenever the gradients of density and pressure are not parallel, you tend to get swirliness. This might be, if it were actual weather, the generator of a small tornado or region of vorticity in the atmosphere, for example. So we think there's a thresholdless baroclinic instability. So that's what we think is happening, but I haven't gotten to the point yet of varying the viscosity and making it a little smaller, the viscosity of the fluid on which this colony is resting. And that's an interesting thing to do, because what it will do is make the vortex ring more intense. And I want to spend the last couple of slides just showing you what happens when we drop the viscosity down from 600 Pascal seconds to roughly 450. And so let's suppose I drop it down to 500 first. So I'll drop it down to 500. And this is that experiment which produced that strange lizard shape earlier. But now we're at a lower viscosity. The fluid mechanics is more intense. You can see that some of the yeast are leaving the mothership. Again, they don't have flagella. They're not swimming per se. They're, in a sense, surfing on this submerged vortex ring. And if I drop the viscosity down to 450 Pascal seconds, so that's about 75% of what I showed you in the initial experiment, I get something like this. You can see that the colony sort of explodes. And by having an intense vortex ring underneath, it can now colonize the entire Petri dish in maybe 18 hours or so, about 4 times faster than it would if it didn't have, than it would on a hard agar plate, because it's now leveraging the fluid mechanics. And notice that these fingering instabilities, which to me look a little bit, some of it, the physics looks a little bit like Rayleigh plateau instability, that have a profound effect on the population genetics. So we have actually 3 different genotypes here. They're all approximately neutral, but you end up with yellow fingers, blue fingers, and some of these fingers are a mixture of genotypes. So it's affecting the Darwinian outcome associated with these number fluctuations in the different genotypes.
So the single color means that a single cell was detached and start to grow?
Oh, you're saying that do we think these instabilities are the result of a single cell?
Yes,
because of a single color.
Yeah,
possibly. I mean, here it's all, but don't forget this one has already made a clump of blue cells, and so anything coming from this would be all blue. So this probably happens after the genetic demixing at the surface. So sometimes it can be a single cell, but it could also be a clump of cells that is having this instability. It's an interesting variant areas of fluid mechanics because, as you know, these are alive. And so down here is a very nice white light picture of one of these fingers coming out. Now here we have pinch off. You know, this is the kind of thing that Sid Nagel and Michael Brenner used to be interested in when you have water dripping out of a faucet. And, you know, is there some, okay, here's the pinch off. It sort of looks like beginnings of a Rayleigh plateau instability. But unlike conventional such things, these are alive. And the cells are vigorously reproducing. They don't quite succeed in preventing the pinch off. But to me, it's a very interesting arena in which to rethink traditional fluid mechanics problems like pinch off and Rayleigh plateau in the presence of something that's alive. And then the last thing I wanted to show you, I guess we did start 5 minutes late, so I'm roughly on time, is some understanding of what's going on. What is the source of these fingers? So these cells are not actually atoms, of course. They are about 10,000 or 50,000 times bigger. They're 5 microns across. So you might wonder if the discreteness of these cells isn't playing a role. Because if the divergence of if the velocity, the stretching velocity field due to the submerged vortex ring that I was talking about is too strong, it's going to thin out the colony to the point where it gets to be less than 1 cell thick. And so this is a way of expanding radially with all these gaps that takes that into account. So it's possible that it's not standard fluid mechanics of atoms, small atoms and molecules, but something that could also be related to the discreteness. So here's a simple theoretical model of what happens to the thickness of the interface and its radius. If we look at the what's going on with this velocity field that I argued was linear in R and radially outward, it has both the colony pushing and the vortex ring are taken into account in this sort of quasi two-dimensional thing. We have write down an equation for the height of the colony. Here's the advection of the cells, of the height of the colony, 100 microns thick or something like that. There's some diffusion that tends to make the height uniform. And then here's a logistic equation for the height that says the height wants to ultimately saturate at the metabolically limited height H0. And if you solve this equation, it's sort of like a Fisher equation in a way, but it has advection, very strong advection in it. And look at what happens to the height and the radius. For this model, you can show, because the nutrients are going in everywhere beneath the colony, aided presumably by this vortex ring, that the radius is increasing exponentially in time. So this exponential increase is associated with this alpha that comes from the submerged vortex ring. It stretches out the colony. The colony is trying to accommodate that stretching by vigorously reproducing. And the compromise between that stretching and the reproduction embodied in this logistics stuff here is to produce a height that obeys this equation. So if alpha, if the stretching isn't too big compared to the reproduction rate mu, the colony thins out, but it thins out to a finite value given by h naught times 1 minus alpha over mu over long times. But if mu is less than alpha, we get what we call a thinning catastrophe, and it will thin out to 0. But of course, it won't really be 0 because these are finite 5 micron sized objects. They can't be thinner than 5 microns. So something has to happen there, might be related to this fingering instability. Here's a numerical solution of that equation. If there's no stretching, then we get one of the Fisher waves that Yuriv mentioned. These are equal times, and so you can see that that they're equally spaced, so it's a constant velocity Fisher wave. But as soon as I start getting that stretching, as soon as the metabolism kicks in, then the height drops down, and the radius now is starting to increase exponentially fast, the height gets lower and lower, and then finally, when mu minus alpha changes sign, you get this thinning catastrophe. Here it thins out a little bit. Here it thins out catastrophically. That might possibly be related to these fingers. We do not really know the explanation for sure. Let me end with taking the viscosity now to even smaller values, 8 times smaller than what we started with, 85 Pascal seconds. And now the colony can take over the plate in less than a day, as before. But once now the submerged vortex ring is even more vigorous. And so what we find is shown in this movie. The colony no longer behaves like a liquid, but rather like a solid subject to fracture mechanics. Fracture mechanics with growing cells, however. In fact, to me, this looks a little bit like plate tectonics. There's Africa, there's South America. If you zoom in and look at white light, look at this fracture process. Now the colony is behaving like a solid, not a liquid. at this, it's a viscoelastic medium. These tendrils are growing as it tries to fracture. So that's another kind of new area to think about, fracture mechanics with living material where the cells are growing. And maybe other people have thought about it, but I haven't seen it done in this microbiological context. So,
does it break where the boundaries between the different strains are located preferentially?
Very interesting question. As far as we can tell, no. What we're doing now is just competing two different colors, let me run this again, that are neutral with respect to each other. And so if you didn't have fluorescent excitation and these two strains, the two different colors passed in the night, they would think they were identical. So there doesn't seem to be any of that going on here. However, were we to redo this experiment with those antagonistic strains, where the yeast try to poison each other, then we might indeed get fracture along these genetic boundaries. So the genetic boundaries don't seem to know about the actual fracture fault lines, but the fault lines clearly have a profound effect on the population genetics, as you can see here. Obviously, the line tension of these microcolonies, or these smaller colonies, is not big enough to make this smooth on the time scale of the experiment. It's viscoelastic. We've now reached a stage where it doesn't flow while it's growing fast enough to let a real line tension come into effect. That's a very interesting question, but we should redo this with antagonistic yeast and see if what happens. Absolutely. So here's a picture which, similar to the one I started with, here are all these continents that have fractured. And of course, if the original continent had a submerged vortex ring below it due to metabolism, then each one of these continents presumably has a submerged vortex ring as well. And you can see these yeast fragments collecting on the midlines between the continents, suggesting that there are downwellings here. The yeast don't follow it down because of the capillary forces. So we're sort of back to where I started, where we have effectively 2D compressible flow in the surface, and you get these accumulation lines of yeast. And with that, I will stop and thank you for your attention. Sorry to run over. Happy to answer any further questions.
Thank you.
Thank you very much, David, for a great talk. Questions, please.
I have a question. Can I ask?
Yes, please.
So did you look on the population genetics within these flows? I mean, because of the stress, presumably both the division time and the mutation rate will be affected by stresses. Mm-hmm. Is there an effect that you know on
well, so that's a very interesting question, and I guess it sort of bears on tensegrity and ideas about how strains can affect mutation rates and reproduction times. So we haven't measured anything in that direction. It would be interesting to try, and we haven't done any theory, but it sounds like a cool problem. I just haven't done it. We could, presumably, and indeed, maybe this is your original suggestion, go back to this fracture thing and sort of look at close to a fracture surface, about when the fracture is happening, say right around here, and see whether the cell reproduction rates are influenced by the stresses.
So you can collect organisms from different continents and let them compete with each other or something.
Yep. So we haven't done that. We should, and we could. We have, in the original range expansions, when we had the demixing into the different colors, but the colony retained a circular shape, I did do what you said. I collected colonies from the different sectors, in that case, not continents, and recompeted them. And the conclusion there was that there hadn't been any mutations on the time scale of the experiment. And these are strains of yeast that are supposed to be slow mutators, but maybe that somehow changed by...
Yeah, but you have huge stresses here.
Absolutely.
And speed up everything.
Right. No, that would be a good thing to look at. And, you know, you could also redo the experiment, not with a 50-50 concentration of yeast, but with a 99% one color and 1% the other color, and maybe have the dilute fraction as a kind of set of tracer particles and sort of see if the ones that, the minority that were close to these fracture surfaces had a significant change in their reproduction time. That would be a fun thing to look at, I agree.
Thank you, fascinating.
Okay, there is another question in the chat by Andre from Poland, who has some issues with the mathematical formulation. So since his microphone doesn't work, I will deliver the question. Yeah, he says, if the vector, maybe go a few slides back.
Okay. So
you had the part, I guess, with the black background.
yeah, okay, good, I can do that. So I think it's this one, maybe?
Yes, I guess. So Andre asks, is the vector symbol over nabla meaningful, or is it just notation issue? And
okay,
still normal nabla?
Okay, so let's see. Let me just go and make sure I don't think. It's the usual nabla, and there shouldn't be a vector symbol over here, over this one. There should be a vector symbol here, a vector symbol here, and we just didn't write them, so I apologize. Yeah.
Other questions? Okay, I think the talk was perfectly clear.
Thank you very much.
So I suggest we thank again David. Thank you very much, David.
Thank you very much.
Thank you.
Thanks for all the great questions. It's very nice to share this research with you.
Okay, so bye everyone. We'll see you in 2 weeks.
Okay, bye. See you. Take care. Stay safe. Bye.
Bye-bye.
Bye.